Welcome to Brain Ponderings, your window into the fascinating world of research with unfiltered conversations with experts at various subdisciplines within neuroscience, psychology, etc. Today, my guest is Professor Diego Mendoza-Halliday. He's assistant professor of neuroscience at the Center for Neuroscience at the University of Pittsburgh. He also, understand, as an appointment at Carnegie Mellon Neuroscience Institute. Carnegie Mellon's there, Pittsburgh. It's got a strong reputation, at least my understanding, strong reputation initially, like in engineering, but now they're getting into like brain engineering, I guess. So welcome, Diego.
Thank you, Mark. It's great to be here.
And so the topic, what the reason I invited Diego to join the podcast is he's doing some fascinating work, a lot in his postdoc, and now he's extending that in his own lab at Pittsburgh on some kind of fundamental aspects of how our brain keeps functioning in real time and over periods of minutes, I'd say, and keeps everything organized, being able to focus on what you're doing, but still remember where you are in the task you're doing. It's kind of working memory. And he's using some cutting edge electrophysiological techniques and even some really fancy optogenetics. I may have him talk about that. It's early days on that to get it really cause and effect, because there's a huge amount of data, for example, on functional brain imaging, uh, but it's very crude. It's in many respects, both spatially and temporally. You can't really, with functional brain imaging, looks what's going on in real time in not only in individual neurons, but even groups of hundreds or thousands of neurons. Okay, so before you get into the science, Diego, what's your background? Where did you grow up? How did you get interested in science and then brain science?
I was born and raised in Bogota, Colombia, the capital of the country.
Uh,
Uh, Huge city, and it was an interesting time in my life. Happy childhood. Lots of, a yard to discover things and explore animals and things. I think that got me a little
oh,
into biology. We were doing a lot of camping and hiking and so on, and looking at nature was pretty common in my life. So that, and I think my parents, especially my dad's, interest in these things, despite not being a scientist himself. Making, doing experiments or engineering a little steamboat in the bathtub, or things like that, really got me into it. And But I think my interest in neuroscience specifically, where my interest in biology narrowed down to neuroscience, was later on in high school when I began to be fascinated by the idea that I could imagine explanations to, even if they were wrong, explanations to many things that I saw around me, with the help of a lot of theory that I was learning in biology and chemistry and physics and whatnot. Um, but there was one question that none of those classes
yeah,
and none of my intuition could actually seem to find an answer to. And that was the question of why we are actually conscious. That was something that really fascinated me. And I was aware from very early on that was probably going to be a question not to be answered in my lifetime. And yet I thought, I want to be part of it. Whatever we have to do, I'll do it. It's fascinating to find something that breaks with the idea that everything can be explained by or seems to break with the idea that everything can be explained by science. And I knew, and I still know, there is an answer there that doesn't require some sort of duality, or some additional science that is different from the others. I feel like we were heading there. We just don't have the probably the technology to address these questions, because they require um, understanding the complexity of our brain, which is just still out of our hands despite trying our best.
You
But
know,
we're here, we're trying.
I've often thought it'd be good for high schools to have at least like what we call in the United States advanced placement neuroscience or something,
Absolutely.
an actual course on neuroscience because there's so much in neuroscience that people can even apply to their own daily lives.
Definitely. And there's much more now than there was when I was growing
Yeah, yeah,
up. My fascination was completely self-motivated. I knew nothing about the brain.
yeah.
I thought it was a mass of gelatin full of neurons with completely disorganized structure. And I thought, how are we ever going to figure out how that thing works if there's all these wires that are intermingled between billions of neurons with no structure? I later found out that that is not true, that it is a massive gelatin with billions of neurons, but there's a lot of structure and fascinating structure that's going to help us. Yeah, so
Yeah.
that changed my mind a little.
Okay, and then did you go to undergraduate in Columbia or in the United States?
None of the above, actually.
Oh,
I left for Canada. I went to
oh,
Montreal to pursue my undergraduate in biology at McGill University.
ah,
There was no program in neuroscience, undergraduate program in neuroscience at that time.
mm,
But there was a lot of neuroscience going on, just not by name. I had to build my own neuroscience program by taking any neuro or psychology or neurophysiology course I could find in the curriculum, and it did pretty well. I really enjoyed my time, despite freezing myself a few times. Enjoyed my time in Montreal studying that. So much that I actually stuck around for my PhD there, and I stayed. I changed from the biology department to the first psychology department, and then the physiology department to study cognitive neurophysiology. And that's where I got into what I am doing right now, studying cognitive processes, but at the level of the underlying neuronal mechanisms in primates.
and okay, so you learned electrophysiology there, right? And
Yes,
and that was in non-human primates or in rodents? What was that in?
that was in non-human primates.
Yeah, yeah.
Yeah, so
Okay,
by far the best model of human cognition.
and what were your major findings there from your studies? What were you doing, and what was your main?
In my PhD, to summarize that, if I could, you know, put it down to one sentence, I'd say I think what I would take out of that work was the finding that um, working memory representations, the information that we are holding in mind temporarily and actively in our heads, moment to moment, that working memory for visual information um, is, first of all, not only in the prefrontal cortex, like many decades ago it was believed, but also not everywhere in the brain, not in every single area, including visual cortex or early visual cortical regions, the first regions of the brain that receive information from the eyes, so to speak, called the primary visual cortex and early visual areas. That it's also not there, but that it sharply emerges. That information sharply emerges in the activity of neurons just past those early stages, what we call middle or mid stages or visual association cortex. And so that sharp boundary between areas in the early visual brain that process the incoming information from the eyes and make sense out of the low level features and colors and orientations and motion direction, everything we need to then build to complex objects and scenes and so on. Those first areas, then we concluded, are not involved, don't have that activity during the period of memorization, right? After a stimulus is gone and we have to hold it in memory.
I see.
But right after this sharp emergence of this what we call sustained activity that goes past the stimulation, the stimulus presentation,
Yeah.
and continues to represent those features that we're holding in mind, was really fascinating. That sharp emergence was what I think I would condense my PhD into.
And what's the role of the hippocampus in all this? Because that, as far as memory goes, at least short-term memory, that the circuits there have been heavily studied, and there has to be some role for them.
Yes, but it's interesting because the role of the hippocampus in long-term memory has been heavily studied, but the role of the hippocampus in working memory
Ah, I see.
is much less understood.
I
And
see.
I think one of the things that is really lacking and that I'm eager to get into is the question of how working memories or short-term memories are consolidated into long-term memories. And what's really difficult there is that studying long-term memory and studying working memory separately have their own established methodologies that work quite well. We can find correlates of these processes in working memory, mainly in the activity of neurons, in long-term memory, mainly in synaptic plasticity and the plasticity of neurons and so on, which is mostly anatomically assessed. The other one's neurophysiologically assessed. But what happens in that middle where memories,
Yeah.
let's say short-term memories,
Yeah.
become less active and now start to be consolidated into long-term memories is still quite a mystery
Yeah.
for the most part, and it's a really exciting one to get into. So I don't know about the hippocampus. I think that the hippocampus will likely play a very strong role in that kind of process of turning one type of short-term memory into a
That makes
long-term
sense.
memory. But we still have to get our hands into that.
Yeah, and the classic kind of research on the hippocampus, there's certain individuals that had damage, selective damage to the hippocampus, and then they can actually function. I mean, if they were in the room and stuff, you wouldn't know there's a major problem. But then when they have to remember something for more than a minute or so, they can't do it. Are there examples of humans that have lesions in the brain regions that you're finding are important for this working memory? And do they have just a working memory deficit, or do they have long-term memory deficit?
Right, that's a very interesting question. So starting from the lesions to the hippocampus, that's actually classical work of studying patients with hippocampal um, lesions or hippocampal removal,
Right.
especially
Oh,
bilateral. That
yeah.
led to understanding. This is back to Brenda Milner back at McGill, who actually taught me. I was lucky enough to have her
Oh,
as
wow.
a professor in one of my courses, graduate courses, and what she and others ended up establishing was the idea that if you don't have the hippocampus, you lose the ability to generate new long-term memories while not quite affecting those memories that are already consolidated. And also, very importantly, not really affecting to the same extent the ability to hold things in working memory for short periods of time. So I think the work that Brenda Milner and others did with a classical patient called HM, named HM, was showing that that patient could interact with people, with the experimenters and with the scientists and other people, in a way that demonstrated the ability to retain information during that period of conversation. Yet these researchers could come back a couple of days later to
Yeah.
reassess the patient, and the patient would not remember them.
Yeah.
So that was a really interesting dissociation between the two. Now, going to working memory, what type of lesions
Yeah.
affect working memory? There is a lot of classical work, mostly with lesioning large portions of the prefrontal cortex, which was classically associated with working memory function, I think we're discovering now that things are a little bit more complicated than that. It's not a single area to function relationship like we thought and what we wished. The brain is more complex, but those classical studies show that large lesions to the prefrontal cortex would then impair the ability to retain information from short periods of time, as we call it in working memory. And some other researchers, including Michael Peturias, also at McGill, trained by Brenda Milner, it's a small world, then showed that particular lesions to subregions of the prefrontal cortex could cause effects that were more specific to monitoring information in working memory rather than just holding it. So that started to poke into, oh, well, working memory is not just one thing. There are multiple aspects to it, and then multiple regions might actually contribute to them. But I think we're still, surprisingly, in the early stages of that. And so I think my work, fast forwarding to my postdoctoral work at MIT with Robert Decimon, Bob Decimon, then poked a little bit into that. We were interested then in another question, which was how those aspects of working memory interact with other cognitive functions like attention. And so, yeah, Bob has studied attention for a very long time, has proposed theories of attention that heavily rely on working memory, so heavily that many have even proposed that working memory and attention are just two names for the same underlying general function. And the reason for that confusion, I guess, stems from the fact that in those theories of attention, when we attend top down, we are, I guess, actively attending to something because we're searching for something. Let me give you an example. I'm searching for the red key holder of my keys. I know my key holder is red, so a way to help my search is to load the representation of my key holder that I have in long-term memory and turn into working memory, and turn that representation into what we call an attentional template, which is some sort of a little model that we are trying to match in the world. So I know my key holder is red. Let's bring it down to just the color. My key holder is red, so anything that's red, I'm going to attend to, and it's going to pop up in perception, and it's going to make me more easily find my keys than if I were searching non-specifically. Right, so there is where working memory and attention start to get to interact. Right, the working memory representation of red becomes the attentional template that then is helping me boost the activity of neurons that respond to that feature and making me perceive it more. I don't know if that was a little too.
that makes sense. So the selective attention, you're holding recalling, I guess, some representation of your key holder in this case. But then at the same time, you're filtering out anything else, I guess. Right?
That's right.
Because all this information from your visual fields, it's hitting your retina. And so presumably that's getting back to at least the primary visual cortex. But then somehow it doesn't come to your consciousness.
Exactly. And that's what attention really is, right? You cannot filter all that information that reaches your eyes at the level of your eyes or your optic tract and so on. Everything gets to your brain, but obviously you're not interested in everything at all times. If you're looking for your key holder, you want to ignore everything else and just focus on that. And that's where attention jumps in and says, well, I have a little software. The brain says I have a little software that allows you to take that signal that includes everything that is going into your eyes and just preferentially processing what you're interested in and just ignoring everything else. That's the expense, right? You have to give away something in order to preferentially process something else. That's what attention does. And working memory is at the base of that because we believe it's the function where that you're attending to is standing while you're attending to it. Don't forget that you're looking for your red key holder, right? So it's right there in working memory, guiding attention.
So this
That's
is,
to
as
converge.
you know, fundamentally, this process is evolutionarily, it must be very old because it seems like it's critical for survival and, acquiring
Absolutely.
resources
Absolutely.
or
Well, we don't know if the mechanisms of attention that act in the primate brain, in our brains, are the same, similar, or divergent from other species. That might be that there are other ways, other mechanisms that implement attentional filtering that are different from the ones that we implement as primates and as humans. So that could also be a possibility. But I believe the underlying phenomenon or the underlying concept of filtering information probably leads to similar mechanisms, converging mechanisms. An interesting question that you just kind of sparked in my head is, well, and I'm sure somebody must be studying it somewhere, is how do very intelligent creatures that have nothing to do with us, like or very little to do with us, like octopi or
right.
other very smart animals, filter or do that attentional filtering. It must be very different, homologously different in the sense that they don't have a cortex. Their brain is very different, but it could be something interesting to look into. What are the common mechanisms between attention in an octopus and a primate? But that's just kind of taking a little tangent.
Yeah, so the prefrontal cortex, from a brain development standpoint, it's interesting too because it's generally thought it's one of the latest regions to fully develop. Uh, how early is working memory? I guess it might be hard to do if the person can't, if it's a kid that isn't talking yet, um, but they still have working memory. We have a granddaughter, she's going to be two soon, but for the beginning when she's a year old for sure. I mean, it's amazing how much information they have in their brain and can recall. She's very good at remembering where things are. Like her mom will misplace her keys or something, and you know, Isabel
Absolutely.
will remember where they are.
Oh, absolutely. They have a very capable working memory,
Yeah.
and I say that from firsthand experience with my kids, and from early on. And there are ways to probe that before kids can speak. That is a similar principle to what we use when we study working memory in animals that can't
Ah,
really speak, right? We find other ways, and psychologists particularly are very smart at finding alternative ways to, especially child psychologists that study cognition in these children are very smart at probing these functions without the need for verbal assistance, right? There are ways, even passive ways, where you rely on, for example, a baby's interest in something that's familiar or that just became recently familiar by looking oftentimes more at what is unfamiliar, and that is a way for the
yeah.
baby to tell the experimenter, I remember this and I don't remember that because I'm looking more at that than this, right? So these kind of tricks appear with animals, with primates. We can actually they can learn by playing some basic video games that we design. They can learn to report um, memory for representations of past stimuli, of recent stimuli, without saying a single word, of course. And so there are ways to do that. And we have to obviously be very careful to assess those things properly because these animals, especially monkeys, are can be cheaters, and then they can find their way to cheat the video game and get it right without having to do to do what you're expecting them to do. So we have to be smart in how to probe it. But I think the field has done a pretty good job at designing these paradigms to probe working memory. You present a stimulus, and then you remove it. The monkeys or humans or other animals, even rodents, have been taught to then hold a representation of a particular feature of that stimulus. So it could be a color, it could be a shape, or some other feature, if we're talking about vision. And then after a period of no visual stimulation, then there is some sort of a period of testing where other stimuli come on, and the animal has to then report a match or something different that lets the experimenter know, oh yeah, I was remembering this stimulus feature all throughout that what we call the delay period with no stimulation.
Okay, so you have these te so it's mainly is it a touch screen based? Are they actually pointing to things on the touch screen?
We are actually now doing this kind of in-cage training where the animals, just from their home, just like humans learned to work from home recently, we're actually also extrapolating that, and the monkeys can work from home and just play video games at home without actually going anywhere, with these touch screens that we place on their home cages, and they can play these things by touching or with a joystick as well. But they also do that with different ways, by looking at things. Right, their eyes can be a way um, to report their responses. We can track their eyes, or they can use a
Ah,
joystick or buttons or so on. There
ah.
may
Oh, I see. Yeah, tracking the eyes, you can do that really precisely so you know exactly what object they're looking at or selectively attending to, I guess is one
or
way.
even used to report, right? If an animal is told, well, look at one of these stimuli that is going to look like the one we showed you, 10 seconds ago, that is a way for them to report, oh, yes, this one looks like that one. Therefore, I was remembering that one for 10 seconds.
Now, when I was at NIH, we had used touch screens in mice. You can train a mouse for food reward to touch icons on the screen, and we were doing something called spatial pattern separation, where you first they learn to touch one big object, say a square on the screen, get a food reward. Then you put another one up at a big distance between the two, and can train it to touch one of those. But then when you move the objects closer together, they have more trouble discriminating.
Right, you do start noticing differences in cognitive ability between rodents and primates and so on when
Yeah,
you
yeah.
start pushing the tasks to their limits. And that's where primates jump in to kind of become a better model for certain cognitive processes that require complex tasks.
Okay, so in these tasks you're having the monkeys do to evaluate their working memory, their attention, um, so you have put electrodes in their brain. Can you talk about kind of what are these electrodes and how many neurons can you record from? And then go ahead and start talking about what you're finding in terms of, say, when neurons in certain part of the prefrontal cortex are active in relation to the, you know, different, I guess, steps you'd say in working memory, that uh, scenario.
We could It is possible to record from neurons, even in human beings,
Ah,
which is
okay.
really exciting.
Yeah.
There are patients with implants for clinical purposes, and so the technology to record activity from different neurons in different brain areas is quite well established in, all the way up to humans, and in many animal species and so on. And that allows us then to take all of these cognitive processes that we've been talking about and understand what is the underlying mechanism, how neurons actually do it. So in my studies, then I record from different areas of the cortex mostly, um, while these animals perform tasks that involve working memory, some of their attention, and in one particular recent study that we published last year, one in which the animals then hold something in working memory, through a period of several seconds, and then use that information to guide themselves attending to that feature they're holding and ignoring different features, other features, so to speak. So in our case, we used motion directions, but you can use colors, you can use shapes, you can use whatnot. It's the same example as looking for your red key holder. In this case, my key holder is a motion direction. So we used stimuli that with dots that move across the screen with a particular motion direction, and the animals can remember that direction of motion. And you can remove that stimulus for several seconds and then present another set of stimuli that include not only that direction of motion, but another one, let's say one moving in the opposite direction. And the animals can then use that working memory representation to guide attention towards the I guess the stimulus that matches the previously seen direction of motion and ignore the one with the opposite motion direction. That's what we call selective attention. In order to then find a little change in the speed of dots somewhere in the screen. So it's almost like you're looking for a shooting star. But now I'm telling you, well, although those stars that are moving, because there's dots moving the screen, the shooting star is going to happen in this direction, but not in that one. So you can actually just focus on that direction, ignore the other one. And so that's how the task went. So using a working memory representation for a motion direction to then guide attention selectively to better perceive something. So that task was interesting because it was important because it had a working memory component, right? Remembering this motion direction for 3 seconds, over 3 seconds. And now the stimuli come up and you have to now use that memory as an attentional template to guide attention to that stimulus. So we had a working memory period in the task, no stimulation on the screen, and then a period of attention where stimuli come on and you have to use that representation from working memory to guide attention. So an attention period in the task and a working memory period separately from each other. And that was key for dissociating between the two, which I can tell you if that's
okay. Okay, so essentially have so in the what's happening to the activity of neurons in the prefrontal cortex that are active during the, I guess you'd call the recall phase, in relation to the activity of those same neurons during the delay time? Are they still active somewhat during the delay period, or?
Yeah, so in the prefrontal cortex, we also recorded activity in other areas like the parietal cortex, the visual association cortex, and so on, and early visual cortex. But specifically for the prefrontal cortex, to answer your question, what we found was an interesting separation between the signals that we know are related to the working memory process, right, retaining information in the absence of sensory stimulation, and what happens later in the task when the stimuli come up and you have to attend to one and not the other, this kind of attentional selection or attentional modulation. And what we saw was that the neurons that showed the signature of working memory, this kind of sustained activity, encoding the memory for the motion direction, oftentimes were not the same as those that showed this attentional modulation that is a signature of attention at the level of neuronal activity. So the signature is that when you attend to something that contains the feature that a neuron likes, right? If a neuron likes motion upwards and you're attending to motion upwards, then that neuron is going to be boosted in its activity. It's almost like a DJ is, turning up the volume for those neurons that whose feature preference matches what you're attending to, and then turning down the volume for neurons that prefer the opposite feature, right? Giving more of a voice to those that matter, or that's I guess the classical theory of attention in a few words. And so that particular phenomenon of this modulation and activity based on what you're attending to, that phenomenon was present oftentimes in different neurons than the phenomenon of work related to working memory. And that was interesting because, as I said before, we had oftentimes associated the two and said, well, working memory supports attention. They're almost basically the same function. Whatever you have in working memory is what you're attending to. And so the working memory and attention might even be the same function. But here we are observing that the signatures, the neuronal signatures of those two cognitive processes, working memory and attention, were present for the most part in different neuronal
Yeah.
populations.
And now, throughout the cerebral cortex, approximately 90, maybe a little over 90% of the neurons are excitatory glutamatergic, use glutamate. And the remaining neurons are GABAergic inhibitory. So when you're recording, are you recording only from excitatory neurons or also from the inhibitory neurons? And how do you, if the latter, how do you parse out? Because if an inhibitory neuron is active, that means there's probably suppression of activity of other neurons.
Right, that's a really interesting question, and one question that I wish we had better methods,
Hmm.
particularly related to private research. Um, what we rely on, we obviously are interested in how different neuron types contribute to the circuitry that is
Right.
carrying out these functions in prefrontal cortex and in other areas. Unfortunately, the difficulty is that the way we are recording the activity of these neurons, listening to the spiking activity of these neurons, is in a way that makes us mostly blind to who's talking. We're
Hmm.
just basically going in with our eyes closed and our ears open and
Hmm.
listening, but we can't really tell too well who is who.
Hmm.
So we basically record a lot of voices, right? What individual neurons are saying, and that is fascinating, right? To listen to individual neurons separately, and now hundreds of them with new technologies that are appearing, hundreds and I think thousands of them at the same time is fascinating. That's almost paramount or essential to understanding these functions because they're not really single neuron based. They're based on populations of hundreds of thousands of millions of neurons. But the one catch is that so far those basic classical methodologies are still not quite there in letting us know what neuron types we are
Yeah.
recording from. There are ways to more or less distinguish between them. So the voices, it's almost like you're going blind into a cocktail party and you want to know who, I don't know, who's a man and who's a woman. And you're just hearing the voices. Well, the voices sound different. And so even not by looking, you could perhaps dissociate between them. The problem is that here we're not talking about such a good dissociation. The voices of neurons that are, for example, excitatory, pyramidal, glutamatergic, and those inhibitory neurons that you were talking about, the voices are not always clearly distinguishable.
I see.
We do think that the what we call the spike waveform or the shape of those action potentials that different neurons fire have some properties that make them differently shaped, let's say, between them. But so far, those methods have been a little bit flimsy. It's not perfectly accurate at dissociating between those types of neurons. So we have some I guess some hints to what those different neuron types might do, but I feel like that evidence is still a little weak, and that we need some new technology to come in and allow us to then tag neurons of different types while we listen to them, and then be able to determine if they are what neuron types they are. And speaking of neuron types, there are so many neuron types within those types that you talked about, and others. There are people keep talking about more and more and more dissociation between neuron types. So that is the dream eventually. Unfortunately, at least in work with primates, we're not quite there. So
Yeah.
I have to disappoint you in that sense.
Okay. Now, if I were to take a look at a slice of brain, or if someone was to give me a slice of brain, I didn't know which brain, region of cerebral cortex it came from, prefrontal, parietal, occipital cortex, and the neurons were stained on the section. And they asked me, which brain region is this from? I'd say, I don't know. It's because the organization of the neurons in the cerebral cortex is very similar. There's these different layers, and you've been interested in looking at that. One of your, I think your most recent paper on PubMed anyway, is this one on spectro-laminar motif of focal field potentials. Can you talk about that? Because that doesn't that have to do with this, uh, different layers of neurons in the cerebral cortex?
Right. Yeah. So to get back to your initial introduction to the topic, yeah, it's true that there's a fascinating similarity in the anatomical structure of the cerebral cortex between different areas that carry out very different functions. So when you look at the anatomical slices of those neurons to see what it looks like, the neurons are actually layered.
Yeah.
And one thing that is really fascinating is that the number of layers is quite preserved. So there's a motif of 6 layers that are formed by a particular configuration of neuron types and neuron shapes and so on, and connectivity and so on. And so the laminar structure is actually apparent to the eye when you look at it in a microscope. It's very obvious. Staining obviously helps even more. But one thing that I would say is they are not the same. So an expert could tell the difference between, let's say, the primary visual cortex
Ah.
section and the prefrontal section, because they do differ anatomically in the makeup of that motif, so to
Hmm.
speak, where the different layers have different sizes depending on where you are in the brain. So that's really a fascinating thing. There seem to be differences. So a common motif all over the cortex that has what we believe is a canonical computation, a canonical circuit that performs a canonical computation, yet differences in that motif, slight differences that don't part away from that 6-layer motif, but modify it, can then allow, and with different connectivity of those areas with others, can allow an area to do something completely different from the other. That's something that's fascinating. The same circuitry or very similar circuitry, or this canonical motif performing very different functions. So we have similarity on one hand, but difference in the other hand. That's kind of I guess the preamble or the introduction to this. But the problem with that is that you can look at the anatomy, but to understand how the brain actually works, you have to see it at work.
Yeah.
And anatomy doesn't tell you that. It doesn't tell you how neurons are actually working. So then that's where electrophysiologists like myself come in and record from those areas and try to figure out what those neurons and those layers are doing. But it's been quite difficult to do that for a few reasons. First of all, as I said, we electrophysiologists have been mostly blind. We go in with our ears, let's say, but we don't quite know exactly where we are, for example, in that motif. And there are some techniques that in some particular areas work really well to allow you to determine where your electrodes were in that particular motif, layered motif. But they don't work always well, and they have a certain, I guess, inaccuracy. And so that is one challenge. The other challenge is that even when we are able to align ourselves well with all the layers and know which layers we're recording from, one challenge we have faced as physiologists, electrophysiologists, is that so far we had the community had mostly failed to find preserved electrophysiological motifs of activity that would align well with that anatomical motif and be as preserved or as ubiquitous as the anatomical motif itself, meaning that you go to any cortical area that has those 6 layers, and then you see the same electrophysiological motif appearing and appearing again. And that so far had not happened. And so what is really exciting about this observation, this discovery that led to the study you were talking about, recently published last year, was that it somehow is the first observation of a physiological motif, like an activity of neurons,
Yeah.
not just the anatomy of neurons, but the activity that we could find is preserved in all of those areas that contain that anatomical motif. So it gave us hope that maybe we can also study this similarity in the motif with by hearing those neurons, and that we can then start to understand what that motif does in terms of function of neurons and how it relates to the functions of the brain, like working memory, attention, visual or auditory processing, emotion, planning, and whatnot. So that is what the excitement.
So Diego, when you say motif, now you're looking, so when people record electroencephalogram, right, you have these like oscillations in different frequency bands, alpha, beta, gamma, et cetera. Gamma's high frequency. And isn't your finding regarding the motif have to do with different frequencies associated with certain layers?
That's right. So what I kept talking about the motif, but I didn't quite say it yet.
Yeah.
So the motif in the activity of neurons comes when we analyze the oscillatory activity of those populations of neurons or those circuits. It is not in the spiking activity that we find it, but the fact that when you record activity in the vicinity of a particular neuronal population, you can catch these kind of oscillations in the voltage. And they are known to be reflective of synchronization between many neurons, kind of activating and deactivating, or I guess having the same phase of oscillation at the same time that aggregates and allows you to observe that oscillation at least a local level, but also very interestingly at a more global level, or even distantly, like what the EEG is, the electroencephalogram, which is those oscillations being so strong and spanning such large populations of neurons that their potentials or their voltage is so strong that they can still cross the entire skull and scalp and be captured by this little sticky electrodes that are touching the scalp, and still you can see those oscillations in different frequencies. So that's how amazing they are, that we knew about them back in the beginning of the first half of the 20th century already, before we could even know the underlying mechanisms. Now we know they come from these local field potentials that are these oscillations between neurons, and those oscillations can happen in different frequencies. Some of them happen in very slow frequencies, frequency bands that we call theta, even alpha and beta, but they can also happen in higher frequencies, which we call gamma. And so to simplify the idea of the motif, what we observe is that when we record those oscillations in different layers simultaneously, then we discovered that the lower frequency bands, particularly the alpha and beta frequencies, were stronger in the deeper layers of the cortex and became weaker as you go more superficially, whereas the higher frequencies, the gamma, were actually stronger in the superficial layers and became weaker and weaker as you go deep. So they have opposite gradients, so to speak, and that creates this fascinating, beautiful motif of the amplitude of oscillations at different frequencies that you can then represent with an image, actually, which we call the spectro-laminar motif. Spectral because it's frequency based, it's across frequencies, and laminar because it's across layers. And that is what we saw. That image of the spectro-laminar motif is what we saw is preserved, strongly preserved across every single cortical
Mm,
area that we have reported from
interesting.
up to 14. I think we've now added a few, maybe we're at 16 areas of it, which was just quite a lot.
So, okay, so I guess one question then maybe talk about optogenetics, which really allows you to get at cause-effect relationships. In other words, you're finding when you have a monkey do certain tasks, you see these changes and, you know, during different phases of the task and so on in activity in neurons in this region. But that doesn't necessarily mean that those particular neurons are critical for working memory, not necessarily, right? It could be an association, but not causally. So, how do you do that? Well, you have to stop the activity in those neurons and then see if it affects the ability of the monkey to perform the task. Um, but before we get to that, and maybe we can finish up on that, I think that's probably something you're working on because I see apparently you've been working, and I don't know if you're continuing this with Carnegie Mellon people or something there, but uh, developing methods to to control selectively the activity of even fairly large populations of neurons in a brain. But I did have one question with the spectro-laminar motif. You mentioned earlier on that not all excitatory neurons are the same, not all inhibitory neurons are the same. There's kind of subtypes. And in the cerebral cortex, the 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 GABAergic inhibitory neurons have been shown to be different in different layers. And I think there's a lot of evidence that these oscillations are strongly controlled by you have activity of an excitatory neuron, but then that activates an inhibitory neuron in this loop. And then that comes back and inhibits, and maybe that's somehow involved in the oscillations. So having said all that, do you think there's a role for different types of inhibitory neurons in determining the different frequencies of oscillations in the two different layers that you found this different?
Yeah, absolutely, Mark. That's a really interesting point because we strongly believe, and when I say we, I guess this brings into the conversation a colleague of mine, close colleague of mine, with whom we started the whole assessment of the spectral-laminar motif across different
Oh,
areas of the
okay.
primate brain. Um, and then extrapolated that work to even other species of primates, including humans, where we found the spectral-laminar motif. So this is actually relevant to humans. It's not something that is just present in primates or animals. But we also found it in mice and so on. That all that work was done in close collaboration with a colleague of mine, Andre Bastos, who I met as a postdoctoral scholar at MIT when he was at MIT and I was at MIT. And since then, he's moved on to start his own lab at Vanderbilt. I've moved on to start my own lab at Pitt, at the University of Pittsburgh. And but we're still very interested in studying different aspects of that spectral-laminar motif, which I call the tip of the iceberg, right? What we've seen is just a little observation. What is underlying that is, figuring out what the mechanisms that generate those oscillations are, what they mean to the brain, why are they there, what are they doing, and this is something that we mostly don't know at all. And so Andrea and I have been working together on some aspects and separately on other aspects. And I do know this is something that he has taken more over. He is, and I'm actually also an author in this work. I've been collaborating in this as well, but it's in his lab that they've been looking into how different neuron types contribute to the generation of this spectral-laminar motif, of the oscillations in different frequencies in different layers. And so far, I mean, it's obviously work in progress, but the first step in that was for Andrew to demonstrate that there is a relationship between the presence of particular inhibitory neuron types and excitatory neuron types, how present they are in different layers, and the presence of different oscillatory components, or these frequencies of oscillation. And so he has demonstrated there is quite a tight correlation between those two things, which points towards the idea that probably the presence of different neuron types in different layers is fundamental to the generation of these rhythms that are layer specific. But this is correlational work, and we still will need to do more work beyond that to demonstrate there's actually a causal role, and it would be really exciting, for example, to be able to inactivate specific neuron types and not others, and see what happens to the rhythmicity of those mechanisms. Optogenetics, hopefully will get us there soon, where you can inactivate specific neuronal types and leave others untouched. And you can do that for different neuron types and then assess their contributions to the rhythms in different layers and so on. But that's work that's work that will need to be done. What was really interesting, though, is not about neuron types, but in general, we did actually use optogenetics at MIT, a particular new opsin in the lab of Guoping Feng that was generated where you basically make neurons, you turn on a light in the cortex, and you make those neurons that have been infected with this opsin, you make them more likely to fire. So now they start activating them themselves. You don't activate them. You just make them more likely to start talking by themselves. And now they start talking as a population. But what was really surprising was that after this conversation starts going between them, you just basically, I guess, triggered the conversation. Now they start talking as a population. Suddenly, after a couple of seconds, you start seeing oscillations happen. So there's an intrinsic tendency for those neuronal populations to start oscillating together. And this oscillation was very exciting because it was induced by optogenetics, not caused directly, right? Because you're not actually turning on the laser and causing the oscillation, but just telling neurons to speak. And by doing that, eventually the conversation leads to oscillation. So that was a fascinating observation that led us to think maybe we can use optogenetics to start studying oscillations by inducing them in certain occasions, or doing that kind of hyperactivation of specific neuron types and seeing what happens to the initiation of different oscillations. But that's work in progress.
So maybe it's possible to enhance working memory performance. Is that?
Some people have claimed that. I haven't really gotten into that. I personally feel that we are not quite there, that if you really want to enhance a function, you really have to master it. Now, you could be lucky. I could say maybe in pharmacology and neuropsychiatry, we have been sometimes lucky with knowing that a particular molecule or a particular drug can actually cure a particular disorder or ameliorate it or solve some symptoms, even without knowing how it does it, right? So you could do that, but I'm more interested in understanding the functioning break to the brain to the point that then we can intelligently design those procedures that would allow us to either return a function, restore a function, or enhance it, and in the best of cases.
And enhance somebody's ability to concentrate and filter out things that are distracting. In modern age, people are doing too much I think too much multitasking and not enough lengthy, like reading something simple, like reading books. People don't read long books anymore. They get all these bits of information, and I think it's important to be able to take the time there's certain limits on how much information we can process and integrate.
Absolutely. That
Right.
pokes into attention and working memory back,
Yeah.
right? The idea that, first of all, attention is used to filter out information to focus on a few items. And the fact that we have that function established in the brain should be a message to us that we should do that. We should focus on a few elements in the outside world at a time and not try to get
Yeah.
to attend to everything at the same time. There's a capacity limitation to attention and a capacity limitation to working memory as well. The fact that if you think of working memory, and this is just a very brute analogy, but if you think of working memory as a desktop where you can place items that you are going to work with and manipulate and transform to come up with solutions to problems or the plan of the day or some
Yeah.
mathematical computation you're doing or any other thought or coming up with a grant. All that is taking space, and that desktop where you're doing that has a limited size, so to speak. And if you start placing more and more items in that desktop, at some point that's going to become messy, and it's going to be more difficult to perform those operations in that little desk. Again, a brute analogy, but that can help us understand why it's important to keep that desktop organized and mostly empty. If you're done with one thing, then you take those items and store them back, or just stop using them, put them in long-term memory, or save them in your computer if there's something that if that's something that you can actually information you can store in your computer, and then move on to the next task one at a time or a couple of at a time. And that's something that I think you poked into it is becoming a challenge in the modern day where there's the internet and the information is just so overwhelming that we just get onto, okay, I guess we give up and say, okay, we're just going to be munching on information at all times and by periods of a minute or two, sometimes 30 seconds as we scroll down our cell phones. That's a problem.
Yeah, it would be interesting to record from the same area in prefrontal cortex when you're having a person do this, go back and forth between and not really focus on one thing, but keep going back and forth.
Yeah, that's something that people have done. So studying
Uh,
working memory capacity and so on, remembering
uh,
multiple items. And there is then definitely plenty of work from other labs showing that there is not only working memory capacity limitations that are well established, well
yeah,
known from psychology, but now people are finding the neuronal correlates of that capacity limitation, saying, look, the neurons that are doing this work stop doing it well after a certain number of items
yeah,
held in memory and so on.
yeah,
But I think going back to you were talking about optogenetics and how that would allow us um, to look into the causality, the causal role
right.
of an area like the prefrontal cortex. And you were, I like that you distinguish between this causal role and just a correlation or an association. You mentioned that distinction between uh, correlation and causation is essential
Yeah,
for us because the brain shows a lot of correlation, right? And a particular area can be fed information from a different area as a say a carbon copy of what's going on in a different area, and that doesn't make it involved in that other process that is going on, and that
yeah,
is a really important. That became a really important I guess, concept when I was looking into the role of the prefrontal cortex in working memory and attention. First of all, we found this dissociation. The neurons that have working memory signals and the neurons that have attentional modulation are, for the most part, different populations. But the more exciting, I guess, finding where we really saw the dissociation between working memory and attention was when we jumped in with optogenetic manipulation of neuronal activity. So we went to the prefrontal cortex, the lateral prefrontal cortex more specifically, the posterior end of the lateral prefrontal cortex more specifically, and a very small region of it, still large, but a subregion. This is not the prefrontal cortex in any way. It's a very small subregion that the lab of Bob Desimone and others have demonstrated is likely involved in the generation of attentional signals that then modulate the activity of visual cortical neurons during this attentional process we've been
Huh.
talking about. So the generation of those signals
Huh.
is likely occurring in this region. Those signals appear earlier than in other areas, and musical inactivation of that area then leads to changes in the modulation of visual cortical neurons. There's a lot of evidence that that points towards this subregion. And so we asked the question: well, if we now go to the subregion and we inactivate it optogenetically, um,
Yeah.
during the attentional period of that task I told you about before. But then we also, in other trials, inactivated during the working memory period instead. So one time in the working memory period, another time in the attention period, and so on. We keep repeating that. Um, can we find can we find differences in the effects of that inactivation of the same neurons that were inactivated, but inactivating, but we're inactivating them in different times of the task. When working memory is involved in maintaining the information in the absence of stimulation, or later on when that representation is then utilized for attentional modulation. And what we found was really interesting. That same population inactivated during the attentional modulation period of the task would lead to an observable and substantial deficit in task performance in these animals. So to
Diego, is
corroborate
that, and that's just inhibiting those neurons and the circuits in the prefrontal cortex on one side of the brain is sufficient?
both sides, importantly,
You
that
have to
thank you
do
for
both
pointing that
sides.
out. That we really want, because I've seen in other studies that unilateral inactivation. So we have, just for the more general audience, we do have two prefrontal cortices,
Right.
one in each hemisphere, and so an activation of one of those hemispheres in the prefrontal cortex could then result in compensatory mechanisms from the other prefrontal cortex and a lack of effects. So I wanted to really prevent that from happening, and so I went beyond what I originally wanted to do to really make sure that effect, that inactivation, occurred bilaterally, to not
Yeah.
give any chance for the brain to compensate. And I think that was also key in observing
Yeah.
this large effect in the performance of this
Yeah.
attention,
Yeah.
working memory based attention
Yeah.
task. But what was most exciting was, okay, we corroborate the role of that region in the prefrontal cortex in attentional modulation, but then the same area inactivated during the delay period failed to impair task performance. And so that's where we saw a beautiful dissociation
Yeah.
between the role of that same subregion of the prefrontal cortex in attention versus working memory. Inactivating it during attention impairs the task performance. Inactivating it during working memory before you even implement attentional modulation does not. That was where
Yeah.
the biggest or the strongest evidence for dissociation between those two cognitive processes, at least at the level of this tiny little subregion of the prefrontal cortex, came in. But even if only one subregion shows this large association, that is already indicative that those two cognitive processes, attention and working memory, are not the same, and
Right.
they have different mechanisms. So that
Yeah.
was exciting.
Yeah, that is exciting. Okay, so what are you doing now there in Pittsburgh, right? You've been there about a year. Hopefully you get hopefully you'll get some funding. I don't know how that's going, but that's
Yeah, it's interesting time.
always the first thing, writing grant proposals, right?
Yeah, so well, I think the most important thing to do when you start your lab is to get things going, right?
Yeah.
You want to start showing preliminary findings, because those are the, let's say, the bait that you throw in the water to get a bigger fish, right? So I use we're now training monkeys in these complex tasks, and on one hand studying what I'll tell you in a second related to working memory, conceived as a bigger concept than typically is conceived. And on the other hand, expanding our understanding of the spectro-laminar motif to assess if we can find it. Basically, an ambitious question, doable though. can we find the spectro-laminar motif in just about every mammal on Earth that has those 6 layers? I think we didn't talk about that, but those 6 layers that you said were present in every single cortical area in the primate brain, in our brain, is actually found in every single mammal on Earth. So you talk about elephants, whales, bats, any mammal that you can think of, which are very divergent, um, they all have the same motif. So now the big question is, well, can we find our spectro-laminar motif in every one of those species? And now we're at it. We're now basically collaborating with a large number of labs all over the world. This is definitely something that could not happen in a single lab. Um, Labs that study in different parts of the world different species of mammals and are interested in studying the layers of the cortex, and so record from layers of the cortex. And what we want to know is, in the recordings from layers, can we still find that spectro-laminar motif that we found in primates and then in humans and mice?
Do you have a telemetry system so they don't have to have wires connected to the electrodes, or how do you, in other words, from a practical standpoint, how are you going to record from a whale or a
Oh, well, I was just talking about what kind of diversity of mammals there are on Earth. I wasn't necessarily saying that we're going to head in that direction. I think we don't have to. I think if we look at the phylogenetic tree of mammals, we can pinpoint key species that are animal models in neuroscience that are
Okay,
become representatives of different phylogenetic branches, let's say.
I
And
see.
if we're able to find the motif in those, then that allows us then to extrapolate or interpolate and say, all right, so then that is probably a motif that is preserved in all mammals. And what does it mean evolutionarily and so on would be really fascinating.
I
That's
had
one.
one of my first podcasts when I started this podcast like 2 years ago was Nikki Clayton from Cambridge. She's at University of Cambridge in England. She's done this fascinating work in crows and uh, corvids, so birds of the crow family, which they're very smart. They can learn tasks very easily, solve mazes, even make tools, use tools.
Now they do little simple math. They can count
Yeah,
things.
they have like theory of mind is clear that they're aware of the presence of another bird and what they're thinking, and but their brain is organized very differently than mammals. So that's kind of a would be an interesting thing to look at in relation to what you're doing, I think, because certainly they must have, I would think, working memory. They do.
Well, they do,
But
yes. The work of
but
Andreas Nieder would point in that direction if you're
but
interested.
their brain's not really organized like ours. They have like just a bunch of different nuclei, collections of neurons, and they're somehow integrated in ways that enable them to do things we can do with seemingly very different brain structure.
That's right. So they don't have that neocortex that we have,
Yeah.
and that 6-layered motif. But they certainly can do so many things. So that leads to a really interesting question. And we are actually trying to extrapolate our, I guess, uh, spectrolaminar framework for brain electrophysiology to even beyond mammals to say, well, can we find motifs of oscillatory activity across layers of other brain structures that are laminated,
Yeah.
and in other species like birds or reptiles and so on?
Yeah.
This could become bigger than we're still able to grasp. So that's to come. I'll leave
Interesting.
that as a trailer for the next.
And then finally, do you have any plans or doing any collaborations in relation to neurological disorders? Are there, say, developmental neurological disorders that uh, have some cognitive problem and there's some implications of your work for that, for those
Yeah,
kind
that's a really interesting topic, aspect of the work. Um, so we obviously study the fundamental aspects of neuroscience. So mainly, we want to start by understanding the functioning brain. That's kind of my
yeah,
my
yeah,
way to work towards understanding disease, and then and then working towards treating disease. And I believe that's the right order
Yeah.
to take.
Yeah.
So we are trying to still understand that, but we have our eyes on where we're heading in terms of how these functions that we are cognitive functions that we are studying relate to neurological psychiatric disorders. So many of these functions, particularly working memory, are impaired in several major neuropsychiatric disorders, including but not limited to schizophrenia and some types of dementia and so on. And so that I think it goes both ways, right? Understanding the fundamental mechanisms of working memory when it is working can then understand, well, what is failing in these patients, in these subjects. But also it goes also the other way around, because failures in these mechanisms can then be used to understand
Yeah.
the functioning brain. Why is it failing when it fails? Can let us know then how it actually works when it works. So I'm very excited to build these collaborations, continue building these collaborations with people studying especially schizophrenia. And one really exciting thing about um, getting into understanding schizophrenia and other disorders that have similar symptoms is one of the symptoms there is the fact that these subjects experience hallucinations.
Right.
The inability, the momentary inability to distinguish reality from imagination. What is a mental representation and what is a real sensory representation? And why that pokes into right into my lab's work is because we are really interested in understanding working memory is the generation of representations, mental representations of what's out in the world and abstract representations of what's not even out in the world. And we believe that the brain has very established mechanisms to maintain a dissociation, a clear separation between what is a representation of something that is presently out in the world,
Yeah.
in front of my eyes, and something that I am generating myself. I can look into a scene and at the same time imagine a different scene, right? And those will not overlap. Obviously, they might be happening in the same kind of visual space, mental visual space at least, but I can clearly dissociate, and I'm not going to start seeing necessarily everything that I'm trying to imagine. There's a clear dissociation, but not in these subjects. And so, my PhD work and on some of my postdoc work poked into how the brain can dissociate between these two. One of the ways there are two mechanisms that might be at work that I poked into in my work. One of them, the fact that I mentioned it before, that working memory representations emerged in mid-level visual areas, not in early visual areas. And so perhaps the brain knows what is a working memory or an imagined representation, a mental representation, because it's not accompanied by activity in these early visual areas or early sensory areas. Could be auditory or other modalities. Whereas if you are seeing something or hearing something from the outside, it it is accompanied not only by the activity of those later areas, but also by the activity of those early areas in the cortex, early visual cortex or early auditory cortex, that are passing on that information. So the presence or absence of activity in those areas can indicate to the brain, this is a sensory representation or this is a mental representation that is not out in the world.
Yeah.
The second, and this is something that came out of my discovery, that working memory representations emerge after the early visual areas. The second mechanism is in the prefrontal cortex. We actually found we had a task where animals would sometimes have to remember a stimulus that disappeared to be able to do a working memory task. But in some occasions, we would leave the stimulus visually present,
Hmm.
so the animal could access it at all times. And we saw neurons in the lateral prefrontal cortex, some neurons that would represent the stimulus when it remained on the screen, but if you remove it, they would shut down. Um, then neurons my favorite, I think, what we call working memory only neurons that were not active when the stimulus was on and remained on. They only woke up and started talking if in those cases in which you remove the stimulus and working memory gets engaged. Then they woke up. And then neurons that we call, I guess, uh, uh, insensitive, that didn't care whether the information was coming from the outside world, the stimulus that remained, or was held in memory, their response and the way they represented the stimulus was identical, and their activity was identical. So invariant to the source of the information. So those three types of neurons told us a lot of things, and one of them is that perhaps that prefrontal circuitry containing these different types of neurons is essential for the brain to distinguish what is out in the world and what is in your mind, and allows you to not fall into seeing hallucinations out of just your mental imagery or mental representations. And that mechanism might be failing somehow in people with schizophrenia. So that's where my interests in understanding the disease really connect to my fundamental work.
Yeah, it would be interesting to do your spectro-laminar analysis in monkeys that have been given a psychedelic, psilocybin or LSD.
Absolutely, yes. Yeah, definitely. To know what happens with that
Yeah.
mechanism. We still don't
And
know, but
also in aging, because just like, you're already some aspects of your cognition are going downhill
oh
already,
yeah, absolutely.
already when you're in your 40s. So in the
I
monkey,
don't want to be a party pooper. I think after 25, everybody starts going down.
and even more for me, but so I was a lab chief at National Institute on Aging for 20 years, and they had a project, Don Ingram, on looking at calorie restriction and lifespan. So they had some rhesus monkeys. I think one got to 40 or something, 40 years old or something. But I assume all of your animals you've done so far are young, fairly young adults. So it would be interesting to look in older animals.
Oh, fascinating question. Absolutely. What happens to that spectro-laminar motif with
Yeah,
aging?
yeah.
It would definitely tell us a lot
So there's two
about.
grants right there that we got, right? The psychedelics, that's one grant, and the aging, that's another
Oh
grant.
yes, and I have others in my working memory right now that you sparked. So go ahead and start writing notes after we finish.
Okay, think this is a good, I think we could talk for another hour and a half, but great, Diego. I really enjoyed talking to you. The time went really fast. It doesn't seem like
Likewise.
we've been talking that long, but we have.
Very
I'll
exciting.
let you get back to work. And exciting time for you. I remember when I was just starting out as an assistant professor at University of Kentucky, 1989. Yeah, it was very exciting. It's a great time. You
Oh, absolutely.
get your lab set
Loving
up.
it.
Yeah.
Loving it. Yes. Thanks again for the invitation, Mark.
Okay.
It's great talking to you.
I look forward to following your work in the future.
Thank you.
Yep. Bye.
Bye bye.